Hey everyone, welcome back. We are going to build on what we did in lecture 12, but in lecture 13 we are going to focus on the physiology of the heart and how it acts as a pump for the rest of the circulatory system. The process that we're going to be discussing overall is called the cardiac cycle. And there are various stages of the cardiac cycle you need to understand before we can go into the physiology of how it works. The cardiac cycle itself is just a series of contraction and relaxation events inside the heart. Remember that systole and diastole are the two terms we use to refer to these two states in the heart. Systole is the act of contraction of the atria or the ventricles. Diastole is the act of relaxation of either the atria or the ventricles. And the chambers of the heart cycle through systole and diastole as the heart pumps blood to the lungs and also out to the body. So let's examine that before we do anything else. We're going to start here, conveniently where it says start. Let me change my pointer to a different color. All right, you can really choose any point in the cardiac cycle to call the start, but we are going to begin with atrial systole. In atrial systole, as the name implies, the atria contract. Now, at this stage of the cycle, they only have a small amount of blood in them, and I'll explain why at the end of the cycle. But here, where we've chosen to begin, the atria are relatively empty, but they do still have some blood in them. When they contract, they squeeze that blood up against the AV valves, which are right here. Remember, those are the atrioventricular valves. The AV valves open because of that pressure, allowing blood to flow down into the ventricles. Okay? The ventricles now contain a full load of blood. Atrial systole then ends and atrial diastole begins. So now we're over here. Okay? Atrial diastole, of course, is the atria beginning to relax. Atrial diastole is actually going to last until the next cardiac cycle begins. So that single act of atrial systole, that's it for the atria until the beginning of the next cycle. Everything that happens from this point forward is going to happen in the ventricles. As atrial systole is ending, as atrial systole is ending, ventricle systole is beginning, and we actually divide ventricle systole into two phases. In the first phase, as the ventricles first start to contract, they generate just enough pressure to force these AV valves closed again. So blood pushes back up against the atrioventricular valves, forcing them to close to prevent that backflow that we want to avoid. As the ventricle continues to contract and pressure builds and builds and builds inside these chambers, eventually the pressure will reach a point that the semilunar valves are triggered and they're finally pushed open, allowing that blood to flow into the arteries, either the pulmonary artery or the aorta. So in the first phase, there's just enough pressure to shut the AV valves. In the second phase, as the contraction progresses, more and more pressure is generated. That triggers the opening of the semilunar valves, allowing the blood to leave the ventricles. Now after that, ventricular diastole begins. Ventricular systole ends after that contraction, relaxation begins, and it's during this stage that blood pressure drops in the body when the ventricles relax. Okay? In late ventricular diastole, once the ventricles have relaxed completely, the entire heart is at rest. Okay? This is the period where both the ventricles and the atria are relaxed. They're in diastole. It's happening right here. Interestingly, at this point, blood flows into the atria because they're relaxed, and then is also allowed to passively flow through the AV valves and fill the ventricles as well. There's no pressure here keeping the valves shut. So as blood is pushed from the lungs or the body back into the atria, because that blood is under pressure, it forces the atria to fill, and then there's a minor pressure difference between the atria and the ventricles, just enough to open up the AV valves and allow most of that blood to passively fill the ventricles. The majority of the ventricle filling that occurs is actually passive, doesn't take energy or contraction. But there's always going to be a little bit of blood left in the atria. Hence the need for us to restart here with atrial systole, to squeeze that last little bit of blood out of the atria, and into the ventricles. And then the system starts over again. Now there's a couple of important measurements that are taken of the cardiac cycle. They're diagnostic, they're important in determining cardiac health. The first is called stroke volume, the second is called cardiac output. Stroke volume is the volume of blood that's ejected by a single ventricle as it contracts. So how much actual blood is forced out of the ventricle when it's undergoing systole. The second metric, cardiac output, it's a little bit bigger picture. It's the total amount of blood pumped by the heart per unit time, usually per minute. Okay? So how do you calculate these things? Interestingly, they're related to each other. You use one to calculate the other, and vice versa. To calculate stroke volume and cardiac output, what you need is the other metric and the heart rate of your patient. So here's some data here. We are looking at a 71 kilogram individual. This is an average weight. For someone of that weight, a reasonable resting heart rate that you might encounter would be 60 beats per minute, BPM, beats per minute. Their resting cardiac output, if measured, would likely be somewhere around 5 liters per minute. So the heart is actually pumping 5 liters of blood every minute in this patient. Okay? So let's say you wanted to calculate the stroke volume for this particular patient using these metrics. The equation you would use is down here. Your stroke volume is equal to your cardiac output divided by that resting heart rate. So for us, for this patient, our cardiac output is 5 liters per minute here. Resting heart rate is 60 beats per minute here. If you divide 5 liters by 60 beats per minute, you're going to get a small number. It's 0.083 liters per beat. Minutes cancels out. Since it's present in both values, it cancels itself out, leaving you with just liters over beats. Okay? You could convert this if you wanted to. You could convert it into milliliters if that's a little easier. This is the equivalent of 83.3 mils per beat. 83.3 milliliters of what? Of blood leaving the ventricle. Okay? Now let's say you've already got your stroke volume for your patient. You have this measured, but you want to calculate their cardiac outputs. We want to do the opposite. You're still going to need their resting heart rate, and you're just going to reorganize this equation. Cardiac output, oops, there we go, didn't mean to cross it out. It's going to be your heart rate multiplied by your stroke volume. So 60 beats per minute times the stroke volume we just calculated, 0.083 liters per beat. That's going to give you an overall cardiac output of 4.98 liters per minute. Now, can we check if that's right? We absolutely can. We were given cardiac output in our original set of data for this patient, right here. Okay. 5 liters per minute versus 4.98 liters per minute. The difference is because of me cutting off some numbers after the decimal. Okay, it's a rounding error. These are functionally the same. All right. What we're going to discuss next is the mechanism that actually drives the rate of a heartbeat and how the chambers organize themselves in terms of which contracts first, which contracts second. All of this is mediated by the nervous system, as you'd expect, and it can be detected and measured by measuring electrical activity in the heart muscle itself. We do this using what's called an electrocardiogram. It's abbreviated as either ECG or EKG. EKG is the older term, ECG is becoming more common. but they both represent the same thing. It's all here in the name. Oops. Electro, meaning electrical, cardio, meaning heart. So you're looking at the electrical impulses inside the heart. Now what's interesting is the cardiac cycle can be broken into 3 different waves of electrical output. So there's 3 different parts of the cardiac cycle. Each one shows up distinctively on an electrocardiogram. Okay? If you look at the readout here, we call this a rhythm strip. The line that shows up on the rhythm strip looks like a series of waves and peaks. Okay? So that's why we call these 3 waves. The first wave is called the P wave. The P wave represents the beginning of the cardiac cycle. It represents the electrical output when the atria are undergoing depolarization. Now remember, depolarization is what triggers electrical surges in neurons. It triggers the start of some sort of change in the cell. Same thing is going to happen here. The P wave is representing the heart as the atria are depolarized, which triggers their contraction. So right here, as a wave of depolarization spreads across the muscle in the atria, it causes them to contract. What comes next is probably the most dramatic part of this curve. It's this big peak here in the middle. Okay. Notice it's got 3 parts. There's a dip at the beginning, represented by the letter Q, a peak up here, represented by the letter R, and then another dip at the end, represented by the letter S. Okay. We call this the QRS wave, encompassing all 3 of those points. The QRS wave shows you where atrial depolarization is ending. So Q, notice how Q dips down low? That should look fairly familiar. That represents a hyperpolarization event. Okay? So the atria are repolarizing, okay, and sometimes they overdo it. This peak of R, it represents the depolarization of the ventricles. Now, the ventricles are going to have to contract much harder than the atria did. It's going to represent a bigger electrical jump because there's going to be a harsher contraction. So this R portion of the QRS wave is going to be high. That's major depolarization that's happening up here. Okay? The end of this wave, represented by S, that's showing you the repolarization of the ventricles. The T wave represents what happens as the ventricles begin to return to their resting state. They're undergoing repolarization and sort of recovering from this electrical barrage that they've just been through. In a healthy heart, where everything's working correctly, your ECG is going to be made up of repeating blocks of this shape. Okay? This is what your EKG results, your ECG results should look like, is this series of waves repeated over and over again. And you see it up here on the rhythm strip. Okay? So there's 1, there's 2, there's 3. So you've got 3 repeats of this rhythm. Now, any variation from this shape, that's going to tell you that there are problems. There's something going on with the heart's rate. Now, electrocardiograms use little measuring devices called electrodes to actually detect these currents from the heart. Electrode placement is really critical for getting accurate measurements. There are a number of different sort of electrode placement systems, depends on the kind of EKG you're using. This is what they look like on an actual patient. This is a pretty old school setup, but they do still use this at some of our local hospitals. A pretty common way to basically triangulate electrical activity in the heart is to place a negative electrode on the right arm, connect that to positive electrodes on the left leg, and connect those to a pair of positive and negative electrodes on the left arm. And this creates a system of leads. A lead consists of 2 connected electrodes. And these leads together are able to pick up on electrical changes happening in the heart muscle. So what happens if you don't get a normal ECG? What happens if your patient's ECG doesn't look like this top row? Well, it could be a number of things. Bradycardia is probably the most common. It's when the heart rate is less than 60 beats per minute. Your heart rate is beating too slowly. You'd be able to tell because these repeating blocks would be more spread out from each other. Okay? There'd be fewer of them happening over this 10-second period because the heart isn't beating fast enough. The opposite of that is tachycardia. Tachycardia is when your heart rate's too high. It's beating more than 100 beats per minute. You would see the opposite of what I just described. These guys would be happening much more frequently. You would see these peaks and these waveforms occurring much more often on this 10-second time interval. There's a couple of different ways that a patient can be tachycardic. They could exhibit flutter tachycardia. This is characterized by really rapid but controlled contractions of the atria or the ventricles. So if you have flutter, you will get a normal shaped waveform. You'll still have a P wave, a QRS wave, and a T wave. Sorry, not on this one. Up here, okay, you'll still have this, but it'll repeat really fast over and over and over again. Now compare that to fibrillation. Fibrillation is another form of tachycardia. It's also going to exhibit rapid beating of the heart, but it's going to be uncontrolled. If you have atrial fibrillation, your ventricles are going to contract normally. So your QRS wave here, that's going to be just normal, just fine. But look at this. These spaces in between, see how messy they are? How many peaks there are? There isn't just your one normal P wave. There's a whole bunch of them. So if we were to zoom in on this, it would look like this. So if we zoomed in here, and then your QRS wave. So clearly this is way, way too many peaks. You're only supposed to have one. Okay? That's how you can tell the atria are fibrillating. They're beating irregularly and too often. Now, ventricle fibrillation, it's going to look a little different. Check this out down here. This is just a hot mess compared to what a normal ECG is supposed to look like. You can't even tell the difference between your P waves and your QRS waves, right? That's probably that's probably a P wave right there, maybe. These are probably QRS waves. But notice how different this looks from our normal or even from our atrial fibrillation ECG. This sort of messiness, where there's obviously too many peaks, there's too many beats going on, and there's no distinctive shapes, this is pretty diagnostic of ventricular fibrillation. Now, fibrillation in general, should be obvious, is not a healthy thing. There are certain ways that it can be corrected. Electrical defibrillation is one of the more common. This is the paddles that they rub together on TV shows and very dramatically stick them in the patient's chest and yell clear, and then they zap the person. It's close to reality. They are going to be zapping your heart using electrical defibrillation. The idea is that a strong electrical impulse into the heart can sometimes re-regulate the myocardial cells, sort of shock them back into synchronicity again. So shock them into beating in sync in a regulated way like they're supposed to. Right. So that's how you measure electrical activity in the heart, but where is it coming from? There's two sources of electrical control of heart activity. The one set of sources that is inside the heart is called intrinsic control sources, and it's performed by what we call a nodal system, or a system of nodes. Extrinsic control comes from outside the heart itself, from other parts of the body. Okay? Intrinsic control, we're going to talk about more on the next slide, but it's mediated by, first and foremost, something called the sinoatrial node, or the SA node. The sinoatrial node, you can see on the diagram right here, so notice it's in the right atria. It's the pacemaker for the heart. So electrical signals from this node is what dictates the pace of your heartbeat and keeps it regular. The atrioventricular node, which is down here, is going to be communicating with the sinoatrial node through what's called, and then also going to be connecting to something called the AV bundle, or the atrioventricular bundle. This is going to connect your atria to your ventricles and help pass messages, electrical messages along. It's sometimes called the bundle of His. I tend to use AV bundle. You can use either one. Bundle branches are what stem from the bundle itself, and these in turn connect to these very fine filaments down here, which are called Purkinje fibers. Now we'll get back to that in a second. That's your intrinsic control system. Now over here, your extrinsic control system, that's found in your ANS, your autonomic nervous system. Both the sympathetic and the parasympathetic systems can help contribute to heart rate. Releases of certain hormones or activating chemicals, think like norepinephrine, adrenaline, that's going to increase your pulse rate, increase circulation, to deliver oxygen more rapidly to your muscles to help prepare you for fight or flight. Okay? You can also, by regulating your breathing, regulating your own body activity, exert some control over your heart rate and help to bring it down. So heart rate can be mediated by either extrinsic or intrinsic factors. I mentioned this in class, but it's been like a month and a half. Let's review what these words mean. Intrinsic, think inside. An intrinsic control factor is something inside the organ itself. Extrinsic, think external. If something is extrinsic, it means it's outside the organ or outside the system. All right, let's revisit this nodal system, this intrinsic system. Okay. All right, we are going to start with the activity of the sinoatrial node. That's step one. The sinoatrial node is going to become electrically active and spread an electrical stimulus across the atria. Okay. That electrical stimulus is going to race across the cellular membranes that make up the muscle tissue of the atria until that electrical impulse reaches the atrioventricular node. Okay, so we go from sinoatrial node to atrioventricular node. Okay. After the message hits the AV node, then the atria contracts. That's what triggers it, is once that message is received by the atrioventricular node, that triggers atrial contraction. Okay. The atrioventricular node is then going to pass that impulse it received onto the AV bundle. Okay. The impulse is going to then travel through the atrioventricular bundle, which is located inside the septum that separates the ventricles from each other. Okay, so the interventricular septum, remember that's that muscular wall that separates the ventricles. That's where you find this AV bundle. Okay. The AV bundle is going to pass the message into its branches, the AV branch, or the bundle branches, and the branches are going to pass the message down into these fine little guys, these little fibers here in the walls of the heart muscle itself. These are your Purkinje fibers. Okay? They are actually the best at relaying electrical signals in the heart. They're probably one of the best series of nerves for relaying electrical signals in the whole body. Okay. When this signal spreads through these fibers completely, that's what then triggers ventricular contraction. And by that point, the atria have entered into diastole and are starting to relax. All right, make sure you can explain this process. Make sure you know the location of all the parts of this nodal system. And yeah, let me know if you have any questions. All right.